In Jesus' name, we are going to pass biology by force. Come, we go through together in the item 2, UACE UNEB Sample Paper, Paper 1, 2026. A paper UNEB sent us to see the paper structure. So this is the item 2. It was coming from what? Also, it's also important to know where it is coming from. It is coming from the topic nutrition in plants, a particular area called gene technology, because we're seeing greenhouses. However, by seeing concepts like we are seeing auxins, gibberellins, and what have you, it means there is also coordination in plants. Good, because a standard item should contain at least 2 learning areas. Now, let's go together. Item 2 was, due to frequent climate conditions that lead to poor crop yields, farmers have been advised to start using greenhouses. Now they define for you. A greenhouse is a house-like structure covered with a transparent, translucent roof and walls that create a controlled environment for growing crops. In other words, a greenhouse, you grow, you have a structure, but this structure here, it is done what? It is made up of either transparent or translucent material. In other words, you grow your crops inside here, What have you done? You've created a microclimate. From the rest of the world, meaning here you can control what is inside there. You can determine carbon dioxide concentration. You can determine the amount of light. You can determine temperatures. You can determine whatever. You can create your own environment here because you've created your own microclimate where you grow your crops. Because of global warming, now experts are advising farmers to start using greenhouses. Another good thing with greenhouses, they can even be used in urban farming where land is limited. Good. Now, to determine the suitable condition to be used in greenhouses, remember you have to determine your own conditions, but which ones are good? Okay. To determine the suitable conditions to be used in the greenhouses, the farmers hired an agricultural officer who carried out trial tomato growing using 4 greenhouses. So for experimentation, of course, greenhouse 1, 2, 3, 4, with the different microclimates inside there. Okay, each greenhouse had different conditions, and the results of the trial are shown in table 2. This was table 2 in the UNEB sample paper, which I have here. Good. Now, for good yields, the agricultural officer recommended that the conditions of the greenhouse should not be kept constant all through. The farmers needed to know how to implement the recommendation. You see, conditions of the greenhouse should not be kept constant all through, meaning they have to be changed over and over, depending on the stage of growth. Now let's look at greenhouse 1, because we need to go to, because here you listen to task A. Analyze the relationship between the conditions of the greenhouse and the growth and the physiological traits of tomatoes. Hello? Analyze the relationship between the conditions of the greenhouse and the growth and the physiological traits of tomatoes. Good. Now, listen, members. We want to analyze the relation between the conditions of the greenhouse. The relationships between the conditions of the greenhouse. Conditions are here. Greenhouse 1, the temperatures are 30 degrees Celsius plus oxygen Z. Greenhouse 2, conditions are 30 degrees Celsius plus gibberellin Z. The difference here is that the temperatures are the same, but the difference comes in the growth substances. Oxygens and gibberellins both are growth substances. But a learner who revised already knows that oxygens promote apical dominance, will always suppress growth of branches, but promote primer going upwards. The apex, whatever as it is, dominating the lateral buds. The apical bud dominates the lateral buds, meaning auxins are opposes branching. Gibberellins promote branching because they promote lateral bud growth. Good. And also a learner who revises knows that the more the branching, the more the leaves of the tomatoes. The more the leaves, the higher the photosynthetic product. Altogether, good. Now, greenhouse 3, temperatures are lower. Room temperature, 25 degrees Celsius plus auxins. Also 4, 25 degrees Celsius plus berries. The difference comes in the growth substance in there. Okay? That's fantastic. Now look. In greenhouse 1, germination rate is moderate. And in 2, germination rate is moderate. There they are similar. In 3, it is also moderate, also similar to these ones here. In 4, germination rate is high. Hope that one can appease everyone. Everyone can be appeased by that. In 4, germination rate is high. Meaning if I grow my tomatoes, I would like them to grow very fast. That's very good. We proceed. Now, photosynthetic rate, micromole carbon dioxide molecules per square meter per second. Here it is high. Carbon dioxide, remember, is a raw material during photosynthesis. It is absorbed. Meaning in greenhouse 1, high carbon dioxide is absorbed. Plant uptake of carbon dioxide is very high. Also greenhouse 2, plant uptake of carbon dioxide is very high. In greenhouse 3 and 4, plant uptake of carbon dioxide is low. Could it be that the low temperatures in greenhouse 3 and 4, could it be that they are somehow inactivating enzymes involved in these photosynthetic pathways? We shall see. We go to number of branches. Greenhouse 1, few branches. Because of auxins, remember, promote apical dominance, hence suppressing lateral buds that would give rise to branches. Greenhouse 2, very many branches. Very many branches in greenhouse 2. Remember, branches comes with a lot of leaves, also high rate of photosynthesis. Okay? Then here, branches are few, hence few leaves, hence low whatever. Here, branches are many. But when you compare very many and many, I would like very many. When you look at these parameters, all of them, greenhouse fund 2 are winning because we will need high rate of germination, meaning at a step of germination. Remember, the officer recommended that conditions of the greenhouse should not be kept constant all through, meaning at the stage of germination, I need to keep 25 degrees Celsius plus gibberellins. It will give me good results of high rate of high germination rate. But when we reach a level of fruiting, I would like greenhouse 2 condition, 30 degrees Celsius plus gibberellins, where they are what? They are very many what? Branches, hence high rate of photosynthesis. Now let's go to our task A. Let's go to our task A, and we'll try to go through it. When you call my number, it is on your bike, I will be responding to you. Only that when you find that it's off, just know that I'm recording some videos, because whenever I'm recording, I put it in flight mode, because I have one whatever until you give me money to buy another phone. Now, let's proceed. Now, task A, analyze the relationship between the conditions of the greenhouse and the growth and the physiological traits of tomatoes. Between the conditions of the greenhouse and the growth and the physiological traits of tomatoes. Good. Now, let's start the analysis. Here, we need to analyze and say, I can begin by saying, I will talk about germination. Germination rate depends on the conditions available, depends on the climatic conditions available in the greenhouse. Germination rate depends on the climatic conditions available in the greenhouse. Available in the greenhouse. Full stop. I can say in greenhouse 1, 2, and 3, the germination rate is moderate. Comma. In greenhouse 4, germination rate is high. Full stop. This is because conditions in greenhouse 4 favor germination due to the temperatures and the growth substances, gibberellins, which promote germination. Full stop. Gibberellins activate amylase enzyme. Gibberellins activate amylase enzyme, which catalyzes hydrolysis of starch into simple sugars, glucose, that is required for the respiration of the embryo and its growth and development into a seedling, hence promoting germination. Full stop. 25 degrees Celsius temperatures favor favor the normal functioning of the amylase enzyme and other enzymes involved in the physiology of a seed to germinate. Okay, full stop. Now look, the other first statement where I said that greenhouse 1, 2, and the other side we said that the rate of germination is dependent upon the conditions in a greenhouse. Full stop. Then you could not say in greenhouse 1, 2, and 3 there is a moderate germination, and greenhouse 4 there is high germination rate. Me writing that, I can award you an A1. Okay? You've noted that, you've observed it. Not every candidate. There may be candidates who may not see it, but I don't think that you. No. You are candidates who went through, have been going through new curriculum. You are very good observers and analyzers. Then you continue and saying that the microclimate in greenhouse 4 of 25 degrees Celsius and the presence of gibberellins instead of auxins favors germination, where gibberellins activates amylase enzyme to catalyze hydrolysis of starch into simple sugars such as glucose to facilitate respiration and hence normal growth of the embryo into the seedling. Okay, into the seedling, and the suitable temperatures, 25 degrees Celsius, also favors the functioning of the enzyme amylase. Okay, me writing such a statement, I can give you an AE. I can award you an AE, which is stronger than A1. Okay, stop. Now let's proceed. Now you've talked about germination. Then you can go and talk about photosynthetic rate as a second paragraph. You can say the photosynthetic rate in greenhouse 1 and 2 is high as that for greenhouse 3 and 4, as that in greenhouse 3 and 4 is low. Me writing that, I will award you an A1. Okay? An A1. Then you can proceed and say this is because the temperatures in greenhouse 1 and 2 this is because the microclimate in greenhouse 1 and 2, 3 degrees Celsius and presence of auxins. This is because the microclimate in greenhouse 1 and 2 favor a high rate of photosynthesis. So you can say 30 degrees Celsius plus auxins activate photosynthetic enzymes such as rubisco to carry out photosynthesis, or to characterize process involved in photosynthesis. Also, 30 degrees Celsius plus gibberellins also activates photosynthetic enzymes such as rubisco to facilitate to catalysis of reactions during photosynthesis. You see, you write about that. There, I can award you an AE because you've brought out that fact there. It means condition temperatures are good for what? For that. You can gonna say the low temperatures in greenhouse 3 and 4, greenhouse 3 and 4. Sorry, the low rate of photosynthesis in greenhouse 3 and 4 is due to temperatures. Now here not oxygens and gibberellins because they are also here, but we're seeing high rate of photosynthesis. So is due to low temperatures, 25 degrees Celsius, that are not in the optimal range where enzymes such as rubisco facilitate a high rate of photosynthesis during the Calvin cycle, like that, if you like. So there you are still you can proceed and put that there. I can even award you an AP. I can also award you an AP. That's very, very important. You would also bring about that fact here having more that immediately. You talk about still the conditions that may not be favoring whatever. So like that. So proceed and go to number of branches. The number of branches are few. We say that is not a good thing. We need many branches because we need many leaves to facilitate a high rate of photosynthesis. Now, greenhouse 2, the branches here are very many. Here you are seeing few, here you are seeing many. But we shall take this one here, and in the long run, this one is the best. So proceed and say, the number of branches in greenhouse 1 is, in greenhouse 1 and 3, number of branches in greenhouse 1 and 3 are few, since auxins, since the growth substance there, auxins, facilitate apical dominance. 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 Hence suppressing growth of lateral buds that will result into branches. Hence, that result into branches. Full stop. Then, me writing that, I would award you an A1. Master, why have you awarded me an A1? By saying the number of branches in greenhouse 1 and 3 are few due to presence of a few. Due to presence, in fact, greenhouse and in fact, let me correct this one here. Because A1 I awarded to trend, I would say the number of branches in greenhouse 1 and 3 are few as compared to those in greenhouse 2 and 4, which are many. We'll stop. There you would say, there you get an A1. Then if you want to say, in greenhouse 1 and 3, branches are few because auxins facilitate apical dominance, or auxins promote apical dominance, thereby suppressing growth of lateral buds and hence fewer branches. There, I award you an AE. Okay? I award you. Remember, we award this coding. We award in layers. The deeper the layer of reasoning, the higher the marks. Deeper the layer of reasoning, the marks. Because A is dominant to A1, AP is dominant to AE, AP is dominant to AP, like that. So, can say that, if you can say, the number of branches in greenhouse 2 and 4 are many because gibberellins promotes growth of lateral buds, suppressing the apical bud, resulting into very many branches forming. There, I award you another layer of score, an AP. I award you an AP there. The more APs, the more APS, the more the winning. Okay, the winning oil. The winning oil. There is a university here, the Great Makere. They normally have that slogan, the winning oil. The winning oil. Then you repeat, you repeat, oil. The winning oil, oil. The losing is Z. Z. The losing is Z, meaning that is very bad. The losing is Z. Like you now, this year, the losing is Z, the winning oil. So when I say the winning oil, you say oil. Okay, let's proceed. Now, from there, you also, another vigilant learner, you can also give us the reason behind the very many. Both are many, but these are very many. Very many, not many. Many, but very many. You can say, the number of branches of tomatoes in greenhouse 2 are very many due to suitable temperatures, 32 degrees Celsius, that favor proper growth and development together with the presence of gibberellins to form very many branches. Such a learner, I can award you an APS. You are an outlier, for you are a candidate of getting an A, and you are a candidate of getting a very good course at campus. I know your course, I know the course you want to do. All biology students, I know your course. Don't lie to me. Everyone will think about that course. Okay, may God act accordingly, basing on his plans. Okay, let's proceed. So with that, you've crushed task A, which was analyze the relationship between the conditions of the greenhouse and the growth and physiological traits of tomatoes. You've already defeated such an item, such a task. Let's go to task B. Justify the recommendation of the agriculture officer and advise the farmers. Justify the recommendation of the agriculture officer and advise the farmers. Recommendation of the agriculture officer and advise. Justify the recommendation of the agriculture officer and advise the farmers. Good. Now, the agriculture officer gave only one recommendation, members. Don't forget, the agriculture officer gave only one recommendation. Only one. Which recommendation? Conditions of the greenhouse should not be kept constant all through. Should not be kept constant. Conditions of the greenhouse should not be kept constant all through. So what does it mean? Let's try to justify this recommendation. Now, from the table, it is very visible. During germination stage, this was the best, high germination. After growth, during photosynthesis, form fruits, what have tomatoes. This is the best, very many branches. The more the branches, the more the tomatoes formed. You see? Because the more the photosynthesis, the more whatever. Remember, the tomatoes are the fruits. So fruiting is very high here due to this combination of 30 degrees Celsius and the growth substance present, the gibberellins. Good. So you need to base on that. We're going to say, changing the conditions of the greenhouse. Changing the conditions of the greenhouse is good, since at different stages of growth and development, tomatoes need different environmental conditions as seen in the table. Full stop. That one is very good. There, I can give you, for example, an E1. You proceed and say, during the germination stage, during the germination stage, the conditions should be like those of greenhouse 4, since they are the ones facilitating a high rate of germination. Then facilitating a high rate of germination, but after germination, conditions should be like those in greenhouse 2, since they facilitate branching, and hence since they facilitate branching, they facilitate formation of a lot of branches, and hence a lot of leaves. Full stop. Then there I can give you an AE. Then you proceed and say, the more the number of branches and hence the more the number of leaves, this results, like in greenhouse 2, results into a high rate of photosynthesis and hence formation of very many fruits, in brackets tomatoes, giving high yield Z. Then there I can proceed and give you an EP from like that. What do you learn from this discussion? Student A and B, all of them will get the answers correct. But president, one of them may have a deeper reasoning and hence we have stronger scores than whatever. But all of them have ticks. None of them has not. All of them have ticks. But one is winning, the other one is somehow not winning very well. So that is the thing. So this long video is, I remember BiologyKingUg. This long video, you find it on YouTube. Of course, if you see it's a short video on TikTok or today, that one to like today, in most cases, like at this time, in most cases, later in the evening, you will see the other video on YouTube. Sometimes I don't upload them as premium. I normally schedule them after some time, or it may even be I send it to YouTube servers, then it may be put to public tomorrow at a given time. I have set in my YouTube. So with that, thank you very much for listening. Let me wait for particular concerns from this discussion. Put them in the comment section. I will be replying to you very swiftly. I remain Biology King Yugi. May God bless you with success this year.